Good day, everyone. You're welcome to the revision class back in 251, where we are revising amino acid and protein chemistry step by step. Like I said, I decided to break down this revision class so that you will have a program or a unit discussed independently, but reference can only be made to the previous class or the later class for better comprehension and understanding. In today's revision, we'll be talking about the classification of amino acids. We'll see how these amino acids were classified into a different, for easy understanding, for easy knowing of those amino acids. So these amino acids were classified into four groups. The classification based on four groups. So the first is based on structure. The amino acids were classified based on their chemical structure. Amino acids were also classified based on their polarity, ability to interact with aqueous environment and the non-aqueous environment. Their ability to also be produced by a human system is also was also used to classify these amino acids. So and metabolic rate, what happened to these amino acids once they find themselves in the biological system? What happened to them was also used to classify these amino acids. So in today's revision, we will be looking at these four bases of classification and see what happened, why these amino acids were classified this component. Now, starting with the first one, classification of amino acids based on the structure. Based on the structure, they were classified into seven. The first one, we have amino acid with aliphatic side chain. Amino acid with aliphatic side chain were further classified into two. There is an amino acid that does not contain a branch. There are amino acids that contain a branch. Glycine and alanine are the amino acids that do not have a branch. And we can see from the normal structure of an amino acid, we have attached to the alpha carbon hydrogen, we have a glycine. While in methyl group, we have alanine. While attached to the alpha carbon, we have a beta carbon. Attached to the beta carbon, we have a 2-methyl group. This is what we call valine. While in leucine, we have attached to the alpha carbon, we have a beta carbon. Attached to the beta carbon, we have a gamma carbon. Attached to the gamma carbon, we have 2-methyl group in branch format. This is leucine. For isoleucine, we have attached to the alpha carbon, we have a beta carbon. Attached to the beta carbon, we have in branch a methyl group and an ethyl group. This is isoleucine. The next group are amino acid containing hydroxyl group. The amino acid containing hydroxyl group are three. We have serine, threonine, we have tyrosine. In the case of threonine, we have attached to the alpha carbon, we have a beta carbon. Attached to the beta carbon, we have a hydroxyl group. In threonine, we have attached to the alpha carbon, we have a beta carbon. Attached to the beta carbon, we have two groups. A hydroxyl group, as in the case of serine, but in addition to serine, you have a methyl group also attached to the beta carbon that makes serine to be threonine. Then tyrosine. In tyrosine, we say we have attached to the alpha carbon, we have a beta carbon. Attached to the beta carbon, we have a benzyl ring. Attached to the benzyl ring, we have OH group. That made it to be tyrosine. The OH group, like we said, the OH group is what differentiates phenyl alanine from tyrosine. We'll see. Next one is sulfur-containing amino acids. Sulfur-containing amino acids are two. cysteine and methionine. We have explained cysteine already with the formation of disulfide bridges between the two cysteine amino acids. Now, in cysteine, what happened? Attached to the alpha carbon, we have a beta carbon. Attached to the beta carbon, we have sulfur hydryl group, just like we have the hydroxyl group as in the case of serine. Now, in methionine, we have attached to the alpha carbon, we have a beta carbon. Attached to the beta carbon, we have a gamma carbon. Attached to the gamma carbon, we have a thioether linking the gamma carbon and the lambda carbon. We have a thioether. We've explained what is a thioether. We've explained ether. We said what is a thioether from ether. Then the next is acid amino acids and their amides. In acid amino acid and their amides, we have aspartic acid, and the amide of aspartic acid is asparagine. We have glutamic acid, and the amide of glutamic acid is glutamate. Now, aspartic acid is, you have attached to the alpha carbon, you have a beta carbon. Attached to the beta carbon, you have a carboxyl group, another carboxyl group. This is a classical example of dicarboxylic amino acid. Amino acid that has a two carboxyl group. So this is also an example of amino acid that has a negative charge because of the secondary carboxyl group. So in the formation of the amide of aspartic acid, we say an amino group is attached to the secondary carboxyl group of aspartic acid to form what we call an amide. This amide contains the carbonyl carbon, and attached to the carbonyl carbon serving as the gamma carbon, then you have an amino group. Then the next is glutamic acid. Glutamic acid is attached to the alpha carbon, you have a beta carbon. Attached to the beta carbon, you have a gamma carbon. Attached to the gamma carbon, now you have the secondary carboxyl group. Now, this secondary carboxyl group is the one that receives an amino group, leading to formation of an amide of glutamic acid called glutamine. So the glutamine, you have the carboxyl carbon receiving the amino group to convert it to carbonyl carbon, and then you will have the amino group attached to this carbonyl carbon to form the classical of what we call an amide. Now, the next one are the basic amino acids. Basic amino acids are three: lysine, arginine, and histidine. The lysine, we have attached to the alpha carbon, we have the beta carbon. Attached to the beta carbon, you have the gamma carbon. Attached to the gamma carbon, you have the lambda carbon. Attached to the lambda carbon, you have an epsilon carbon. Attached to the epsilon carbon, you have an amino group. That is lysine, or simple as that. Then arginine. In arginine, we say attached to the alpha carbon, you have a beta carbon. Attached to the beta carbon, you have the gamma carbon. Attached to the gamma carbon, you have the lambda carbon. Attached to the lambda carbon, you have a guanidino group. And we said the guanidino group is nothing but a carbon surrounded by three amino groups. Now, the next one is histidine. Histidine, we said attached to the alpha carbon, you have a beta carbon. Attached to the beta carbon, you have an imidazole group. And imidazole group, we say, is a 5-atom, or 5-atom ring, or nitrogen containing 5-atom ring, which is the classical of what we've seen here. And then we have aromatic amino acid, and aromatic amino acid we have attached to the alpha carbon, we have the beta carbon. Attached to the beta carbon, we have the benzyl ring. Then we have the tyrosine, which I've already explained the difference between tyrosine and phenylalanine. And tryptophan, we said in tryptophan, we have attached the alpha carbon, we have a beta carbon. Attached to the beta carbon, we have an indole group. And this indole group is nothing but a benzene attached to a 5 nitrogen containing member ring, which is classical of what we have seen here. And then we have the last one, which we call an amino acid. An amino acid, we said, is an amino acid where we have attached to the alpha carbon, we have the beta carbon. Attached to the beta carbon, we have the gamma carbon. Attached to the gamma carbon, we have the lambda carbon. Now, attached to the lambda carbon, this lambda carbon form a bond with the amino group of that amino acid. That made it to form this kind of pipe member, nitrogen containing pipe member ring molecule called pyrrolidine or that look like a pyrrolidine ring. Now, we'll finish a classification based on chemical property. Now, classification based on nutritional concern. They are classified into three: essential, non-essential, and then conditionally essential. Initially, it was essential and non-essential. Essential are those ones that cannot be produced by the body, which include histidine, isoleucine, lysine, lysine, methionine, phenylalanine, threonine, tryptophan, taurine. You need to eat food that contain these amino acids for this amino acid to be available in your biological system. That is why it's always well to encourage, it's good to always encourage food that contain all the necessary nutrients so that this amino acid can also be found in your diet. If you are taking the food that contain non-essential amino acids, that contain no essential amino acids, you fall into a number of challenges. Sometimes even some challenges that you yourself don't know to know, and the doctor will find it difficult to also know what is actually happening. This is quite a number of symptoms showing. Then you have a non-essential amino acid, those ones that can be produced by the body. But among them, there are some, among the non-essential amino acids, there are some, even though they are produced by the body, but under a several some stress condition, under some certain conditions, maybe when the body actively needs them in high quantity, the body then fails to produce them. So those ones were isolated and then tagged as conditionally non-essential amino acids, which are originally or conditionally essential. Sorry, these are, it's a mistake here, please, you can correct that, conditionally essential. The arginine, cysteine, glutamine, glycine, proline, and tyrosine. Now, the next one is classification based on metabolic rate, where we said some amino acids in the biological system were converted to acetyl-CoA. From acetyl-CoA, they are converted to ketone bodies, while some amino acids can be converted to glucose. So when you take an excess food, this excess food will have to be converted, sorry, excess proteins will have to be converted to amino acid, and these amino acids, because cannot be stored in the body, they have to be excreted out. So for that to be excreted, the easier way is the carbon skeleton of this amino acid can actually be taken and used to produce other things, while the amino group, which are toxics in form of ammonia, will have to be taken out of the body in form of urea, as in urine. They're excreted in form of urine. So these amino acids, some of them can be, when the carbon skeleton is being removed or the ammonia has been removed, then the remaining carbon skeleton will now be used to produce two major molecules. Either acetyl-CoA. Acetyl-CoA can be used for energy production because the body doesn't need that. The acetyl-CoA can actually be used to synthesize what we call ketone bodies. Ketone bodies are also sources of energy that can be stored in the body and then later be converted back to acetyl-CoA and utilized for energy production. Or some carbon skeleton can be used to produce glucose, and the glucose, of course, can be converted to glycogen, and glycogen can be stored in various tissues like kidney and skeletal muscles. Now, there are some amino acids that can also be used to synthesize both glycogen and ketone bodies. So they are in between, they are mixed ketogenic and glycogenic amino acids. We have an example of them we talked about. Then the next classification is based on the polarity, where we said they are classified as charged, negative, positive, non-polar, and polar. So there are charged amino acids, and we say this charged amino acid in a solution can actually interact with the ions that are produced in water, the hydroxy and hydroxonium ion. Likewise, the negatively charged. Likewise, the polar, because of these highly electronegative atoms like oxygen found there. And then we have non-polar amino acid. This non-polar amino acid find it very difficult to actually interact with polar with aqueous environment. To have example of lysine, isoleucine, methionine, tryptophan, etc. So this is basically the classification of amino acid. In our next class, we will be talking about chemical properties or chemical and physical properties of an amino acid. For now, if you have any question, please pass your question to the class and down to me. Your answer will be from me through your class down to you. Thank you.